In the realm of animal reproduction, the distinction between egg-laying, oviparity, and live birth viviparity has long been considered a clear-cut divide. However, recent observations and studies, particularly in reptiles, have revealed a far more complex and nuanced picture. This fascinating area of research not only sheds light on the diversity of reproductive strategies in nature, but also provides valuable insights into the evolution of reproductive methods, including our own. The remarkable case of the Australian three-toed skink. In January 2014, an Australian three-toed skink made headlines in the scientific community by demonstrating an unprecedented reproductive flexibility. This single individual laid 3 eggs, with one hatching several weeks later. Remarkably, about a week after the egg hatching, the same parent gave live birth to another offspring. This extraordinary event marked the first documented case of a single animal producing both eggs and live young in the same reproductive cycle. This occurrence challenged the traditional binary view of animal reproduction and highlighted the existence of a continuum between egg-laying and live birth. It sparked renewed interest in understanding the evolutionary pathways and mechanisms behind these reproductive strategies. The spectrum of reproductive strategies. Traditionally, animal reproduction has been categorized into 2 main strategies. Oviparity, egg-laying. This is the ancestral and most common method among bony animals. Oviparous animals, such as all birds and turtles, lay eggs that develop and hatch externally. This strategy allows for larger numbers of offspring with less physical strain on the parent, but leaves the eggs vulnerable to environmental threats. Viviparity, live birth. This method, where offspring develop inside the parent's body and are born alive, is most famously associated with mammals. It offers greater protection for developing embryos, but typically results in fewer offspring and greater physical demands on the parent. However, the Australian skink case and numerous other examples, particularly among squamates, lizards, and snakes, reveal a spectrum of reproductive strategies that blur the lines between these 2 categories. Egg retention. Some species produce eggs but retain them within the body for extended periods, allowing for more advanced development before laying. This strategy provides some of the protective benefits of viviparity, while still utilizing the egg structure. Ovoviviparity. In this method, eggs develop inside the parent's body, but the embryos rely primarily on yolk for nutrition, similar to external eggs. The parent then gives birth to live young that have developed within egg membranes. Facultative viviparity. Some species can switch between egg-laying and live birth, often in response to environmental conditions. This flexibility allows for adaptation to varying climates and habitats. The evolution of live birth in squamates. The group of reptiles known as squamates, which includes lizards and snakes, provides a unique window into the evolution of reproductive strategies. Unlike mammals, where the transition to live birth occurred once in the distant past, viviparity has evolved independently over 100 times in different squamate lineages. This repeated evolution makes squamates an invaluable model for studying the transition from oviparity to viviparity. Approximately 1/5 of all squamate species are livebearers, including various snakes, skinks, and lizards. This diversity allows researchers to observe different stages of the evolutionary transition to viviparity, from species that merely retain eggs for longer periods to those with fully developed live birth mechanisms. The transition to viviparity in squamates often follows a pattern: increased egg retention, thinning or loss of the eggshell, development of placenta-like structures for embryo nutrition. This gradual process explains why we see such a variety of reproductive strategies among lizards and snakes, including species that can employ both methods. Embryonic nutrition and the evolution of the placenta. One of the key challenges in the evolution of viviparity is how to nourish the developing embryo without the resources provided by an external egg. In many viviparous lizards and snakes, the solution is relatively simple. They retain the yolk while losing other egg components like the shell. The yolk continues to provide nutrition to the embryo inside the parent's body, much as it would in an external egg. However, some live-bearing lizards have evolved more sophisticated methods of embryo nutrition that parallel the placental system seen in mammals. These species have repurposed tissues that originally evolved to support embryos and eggs, such as the yolk sac or the chorioallantois, a gas-exchanging membrane, to create new structures specialized for nourishing embryos internally. This evolutionary innovation is strikingly similar to the development of the placenta in mammals. By studying these transitional forms in lizards and snakes, researchers can gain insights into how our own ancestors may have evolved from egg-laying to live birth, including the development of complex placental structures. Implications for understanding mammalian evolution. The study of reproductive diversity in squamates offers valuable lessons for understanding the evolution of mammalian reproduction. While the transition to viviparity in mammals occurred too far in the past to study directly, the multiple independent evolutions of live birth in lizards and snakes provide observable models of this process. By examining the various stages of this transition in squamates, from simple egg retention to the development of placenta-like structures, researchers can piece together the likely steps that led to the evolution of mammalian viviparity and placentation. This comparative approach helps answer key questions about how embryos are nourished in the absence of external eggs, and how specialized tissues like the placenta evolve. Environmental factors and reproductive flexibility. The ability of some squamate species to switch between oviparity and viviparity, or to exhibit different reproductive modes in different populations, highlights the role of environmental factors in shaping reproductive strategies. This flexibility may be an adaptation to varying climates or habitats, allowing species to optimize their reproductive success under different conditions. For example, viviparity may be advantageous in colder climates where external eggs would be exposed to harsh conditions, while oviparity might be preferred in warmer regions where the risks to eggs are lower and the energy costs of carrying developing young are higher. The ability to switch between these modes, as seen in some lizard species, represents a remarkable evolutionary adaptation to environmental variability. Conclusion: the discovery of the Australian three-toed skink's ability to both lay eggs and give live birth in the same reproductive cycle serves as a powerful reminder of the complexity and diversity of life's reproductive strategies. Far from being a simple dichotomy between egg-laying and live birth, animal reproduction exists on a continuum with numerous intermediate stages and variations. The study of reproductive diversity in lizards and snakes not only illuminates the fascinating evolutionary pathways that have shaped these reptiles, but also provides valuable insights into the broader evolution of reproductive strategies across the animal kingdom. By understanding the mechanisms and factors that drive the transition from oviparity to viviparity in these species, we gain a deeper appreciation for the evolutionary processes that have shaped all forms of life, including our own. As research in this field continues, it promises to yield further insights into the plasticity of reproductive modes, the environmental factors that influence reproductive evolution, and the molecular and physiological mechanisms underlying these transitions. This knowledge not only enriches our understanding of biological diversity, but may also have implications for fields such as conservation biology and reproductive medicine. The blurry line between egg-laying and live birth, so vividly demonstrated by the Australian skink, serves as a testament to the incredible adaptability of life and the continuous nature of evolutionary processes. It reminds us that even fundamental biological categories can be more fluid than we once thought, encouraging us to approach the study of life with open minds and a willingness to challenge established paradigms.